Hi everyone, welcome to today's lecture about the causes of emotional and motivated behavior. This is our third last lecture. We're going to continue the class after this talking about sleep, and then we're going to have a final lecture about learning and memory before our final midterm. Today we're going to talk about a number of topics that are loosely related to the idea of emotion and motivation, including talking about our two chemical senses, our sense of smell and our sense of taste. We'll talk about how our behavior is controlled by circuits in our brain, including our hypothalamus. And we'll talk about emotion a little bit and reward at the end of the lecture. Emotions we can define as the cognitive interpretations that we attribute to our subjective feelings. So you get up and your body feels a certain way, and you attribute certain interpretations to those feelings, like I must be nervous about the upcoming exam. A motivation we can define as a behavior that seems purposeful or goal-directed. So seeing someone stare off into space and scratch their head versus walking to a Subway and ordering a sandwich seem to have different apparent motivations. And we'll talk today about the neuroanatomy of emotion and motivation, of course, which will bring the hypothalamus front and center, as well as the limbic system and the frontal lobes. We'll consider their role. We can think of motivation as, in one way, as a drive for the brain to maintain some state. So a classic experiment in which monkeys were put in a dim, boring room with a very small window they could open and look into the adjoining room. There was nothing interesting in their room, and so things were changing in the other room quite a bit. And they spent a lot of time opening the door and viewing what was on display. And they were even willing to perform tasks like they would for food just to be able to look through the window. And the longer that they went without looking, the more time they spent looking, and the harder they were willing to work to look. And so just the absence of stimulation alone was enough to motivate their brains to search out some sort of stimulus. Here's a picture of those monkeys looking through the window. And so thinking about what circuits those monkeys' brains might be utilizing to motivate them to take that behavior instead of, say, scratching the wall or sitting in the corner is of interest to our research. So scientists have identified circuits in the brain for reward, for instance, that are activated when we find things rewarding, and found that these can be modulated, so they can increase or decrease in their activity due to things like hormones or the chemical senses, as we'll see in a second. And we also have to remind ourselves of the assumption that the main reason, the underlying cause of a thought, a feeling, or an action, like someone going to Subway as opposed to McDonald's, the underlying cause of that decision lies in brain circuits that we're talking about today. One section of this investigation includes evolutionary theory. And so evolutionary psychologists might come up with theories for why humans have evolved and other animals have evolved to have certain traits as opposed to others. And we can imagine environmental influences that acted on not the organisms, but in this case, we can even think about them acting on particular brain circuits or particular patterns of activity in brain circuits to prioritize one behavior as another that led to better reproduction. And so we can think of things like, why does the sight or smell of a bird or mouse trigger killing and stalking by a house cat, even that's well fed? And why do images of the human body stimulate human sexual interest? These are thought to be evolutionary adaptations of the organism to encourage it to be more reproductively fit. There's an idea of something called an innate releasing mechanism. This is a hypothetical kind of set of brain circuits that can detect a specific sensory stimulus, like say smell of a cat, and direct an organism to make a particular action when that happens. A great example, as we'll see on the next page, is the way that a cat can arch its back and puff up its tail at the sign of another cat or another threat. This happens even without any learning, so it's an innate activity that happens due to threat. So the brain has to have this set of normal activities and the set of normal images and stimuli that it can match to trigger the appropriate response. And they're thought to be pre-wired because of how early they're present in animals and humans, but they can be modified over time with experience. So if you present a kitten with this picture A, it will defensively raise its fur and arch its back and hiss. And it starts at about 6 weeks of age, even in cats that have never seen another cat do this. And this jumbled up version of a cat here doesn't evoke the same response. So something about this sensory image is imprinted on a cat's brain to the extent that when it's observed, it can release this motivated response. Humans have something analogous in that our infants can recreate the facial expressions of the humans that they see around them, even at a very early age. And adults around babies are known to often exaggerate their facial expressions as a way to accentuate this communicative channel. And the newborns are too young to be intentionally imitating, so it's thought that they have this similar template matching with a sensory stimulus and their own behavior. These innate releasing mechanisms are pre-wired into the brain but can be modified. And so, for instance, blind children that have never seen can produce the same facial expressions as sighted infants, even though they've never seen them. And in different cultures, for instance, different types of stimuli are thought to be arousing, indicating this modification of IRMs by experience. As we've learned, natural selection favors behaviors and brain circuits that produce those behaviors that prove adaptive for an organism. And adaptive means they're more likely to lead to offspring. And these behaviors in those offspring, therefore, are inherited, and their brain circuits themselves, therefore, can be passed on to future generations. And so animals that reproduce and have healthy offspring are likely to pass on those same brain circuits that made them likely to reproduce, and then make their offspring, therefore, in turn, likely to reproduce. So natural selection we can think of as a selection of brain circuits. Evolutionary psychology, as we've discussed, seeks to apply these principles of natural selection to try to understand the causes of human behavior. And we start by understanding that these behaviors exist because the neural circuits that produce them have been favored by our evolutionary history. But this theory alone can't account for all the things that humans do, like homicide, for example. And evolutionary psychologists then can generate intriguing hypotheses that are admittedly often unfalsifiable, because you can't go back in time and test these things, but they're often quite interesting to think about about how natural selection might have shaped different brain or behaviors. As opposed to thinking about how our ancestral history has led us to behave in certain ways because of what we've inherited from our ancestors, we can also think about alternatively how the environment in which we grew up, in which our brains developed, influenced how they respond currently. And this idea of environmental influences on behavior was really pushed in the 1900s by B.F. Skinner and others as the field of behaviorism, which you've learned about, which suggests that learning plays a strong role in our behaviors in addition to just our evolutionary past. So a reinforcer in this theory is any event that strengthens the behavior that follows. So if you get a bonus at work after making a sale, the idea of the bonus is to try to encourage you to make more of those sales. And our experience, therefore, acts by pairing certain stimuli and situations with reinforcers so that our brain knows in those situations that we're more or less likely to end up on top, to end up having more reward than punishment. So many of our complex behaviors, however, are learned, and learning takes place in the brain, which we already have just covered, has been selected for evolutionary adaptation. So our evolutionary history has prepared us to learn in these ways after we're born. However, there's limits to what we can learn. The environment doesn't always change the brain. A pigeon can learn to peck for food, but it can't learn to peck to escape a mild shock to its feet. So it can make the association between moving its beak and getting food, but not between moving its beak and cessation of a shock, indicating that the brains are not pre-wired to make every association, but only certain associations between stimuli and behaviors. Another good example of the environment influencing our behavior is learned taste aversion, something that many of you may have experienced. This is an acquired, learned association between one specific taste or odor and illness. And it can lead to an aversion to foods that have that same taste or odor in the future, even when an illness isn't experienced. Textbook has a good example of a study in which coyotes were killing sheep in the countryside, and they tried all sorts of things like shooting the coyotes to try to scare them away that weren't working. And instead they tried poisoning the carcasses of the sheep with enough poison to make the coyotes ill but not die. And this was effective at teaching the coyotes to not eat the sheep because of this single learning experience. And some of you may have had the same experience where you've gone out to eat and gotten ill the next day and learned some many months later that you now have an aversion to one of the foods that you ate that night. It's often the food that was most novel that you had at that dinner. It's most salient in your brain. And it doesn't necessarily have to be the food that led to the illness. The illness can be secondary. So this idea of preparedness, the pigeon only learning to peck for food, but not peck to get away from the electricity. We have a predisposition to respond to certain stimuli differently than others, and our brain is pre-wired to make certain associations, like that the sheep is related to illness, but not that the sheep is related to pain. Okay, next we're going to move on and talk about one last sensory system in the brain, our chemical senses of olfaction and taste. chemosignals or chemical signals that play a central role in motivation and emotion, which is why we're covering them in this chapter. They help us to identify members of our groups, to mark territories in animals, to identify our favorite foods and foods that we shouldn't be eating. They help us form associations between odors and tastes and other events that help us to learn to eat in the future and to navigate our environment effectively. Odor and taste play a very fundamental role in emotion and motivation because of these behaviors. Humans can discriminate thousands of different odors, yet we can't really name any one particular odor. That's this vast space of subjective feeling that we can't really attribute labels to, linguistic terms to. We can say things smell like something, but not that they have some particular smell quality unless we're very well trained. Our sense of smell is primarily designed to discriminate whether information is safe or familiar. So is that a smell from edible food? Is that a smell from a friend or a stranger? The scent molecules in the air interact with chemical receptors in our nasal passages. And the lifetime of each of these olfactory receptors is quite brief, about 60 days, and they're constantly being regenerated. The nose at the top of our nasal passage is something called the olfactory epithelium, which is the receptor surface where the olfaction occurs. And each of these receptors that's embedded in this epithelium sends a process ending in about 10 or 20 cilia, little hairs that are embedded into the mucus layer of the nasal passage, called the olfactory mucosa. And the chemicals in the air dissolve into the mucosa to interact with those cilia. And the chemicals cause a metabotropic activation of specific G protein receptors, which we remember are these metabotropic receptors that lead to second messenger processes in the cell. In this case, they open up sodium channels and change the membrane potential of the cell, leading it to change how it's signaling to the next cell in the pathway. So here we can see those odor molecules in the air entering the nasal passage and moving around. We can see them interacting with the nasal mucosa here, in which the cilia are embedded. The cilia you can see are the long end processes like dendrites of these olfactory receptor cells, which we saw can be regenerated every 60 days. Their axons travel through this bone in the top of our nasal passage into our brain itself, where the olfactory bulb is located on the medial surface of the brain. And the axons cluster together in groups, and similar cells that respond to similar chemicals all synapse to the same group of cells called a glomeruli. These glomeruli then connect onto mitral cells, which send axons into the piriform cortex, where olfaction continues to occur. Here we can see the air molecules interacting with those G protein receptors and opening up sodium channels to change the polarization, leading to change in the signal of these glomeruli. During some bad accidents, people's olfactory system can dysfunction. When this bone breaks, these axons can break here, and this can lead to a permanent loss in the sense of smell. So these olfactory receptors project to the olfactory bulb we saw, and on from there, the olfactory bulb's connections move on through the brain and in this case, the smell is our only sensory system where most of the connections don't pass through the thalamus first. There is a thalamic connection that does first project to the orbital frontal cortex, where it's thought that emotional, social, and eating behaviors are organized. So here we can see the olfactory bulb on the medial surface of the frontal lobe, sending projections to the amygdala, to the medial temporal lobe, the entorhinal cortex and piriform cortex. These are areas near the hippocampus that we'll talk about in 2 weeks. And from there, also on to the orbital frontal and thalamus, and to the hypothalamus, which is one of our main focuses today. So this smell information can eventually make its way into the hypothalamus to modulate our motivation as well. Animals have a secondary olfactory system called an accessory olfactory system, which is designed to detect pheromones from their conspecifics, animals of the same species as them. These biochemicals are released by one animal of the species, and they act as chemosignals that can affect the physiology and behavior of the other animal. They're detected by special olfactory receptors known as the vemeronasal organ, which is connected onto the amygdala and the hypothalamus. And there's a small inlet to this vemeronasal organ on the underside of the palate of the cat, which is why the cat can, when it smells the urine of another cat, the cat can make this funny face to open up that pathway for air. The human olfactory system is surprisingly acute and sensitive. We can identify our own odor. We can identify the odor of our kin versus of others. We can identify the odor of our friends versus strangers, all well above chance. We can also identify the odor of all different sorts of foods, obviously, and dangerous, noxious smells as well. Body odors can activate brain regions involving emotional processing in humans. And a stranger's odor, as opposed to a friend's, activates to an increasing extent. By that we mean increases the blood flow and activity in the amygdala and insular cortex, where these emotions are processed. So the processing of olfaction in humans, however, is mostly an unconscious and automatic process that we aren't aware of. Humans have a range of different taste sensitivities, and we are aware of the fact that there's people that are very acute and sensitive to bitter tastes called super tasters. And these people generally don't like foods like Brussels sprouts because of their bitter taste, which they experience much more strongly. You could imagine that these super tasters might have increased expression of certain taste receptors. Children are more responsive to taste than adults. And so by the age of 20, we've actually lost about half of our taste receptors, explaining the picky eaters that we see in children, and also the propensity for increasingly spicy food as we age. Spicy and tasty food. As you know, we have ability to taste 5 different tastes with our tongues. And these taste receptors are actually not just on our tongues, but on the inside and back of our mouths as well. We have receptors for sweet, sour, salty, bitter, and also umami, which is this meaty glutamate taste that makes, for instance, Doritos taste good. The taste of MSG, monosodium glutamate. And you can see there's a myth actually that the taste is distributed on the tongue, such that we have like a salty area and a sweet area of our tongue. That was even shown in textbooks many years ago. This is false, and all the parts of our tongue have taste buds and taste receptors that are arranged like this, where groupings of taste receptors of various ratios of the 5 different types of chemical responses are grouped together in these taste pores. And these taste pores are actually smaller than a taste bud. A taste bud is kind of a physical bump on the tongue, and these taste pores are embedded in those taste buds. And the taste pores have these microvilli on the top that can detect the chemicals in the saliva. And then you can see that these neurotransmitter vesicles can release neurotransmitter onto this axon of cranial nerves 7, 9, and 10 when those microvilli are sufficiently activated by particular taste. We know, for instance, one of the most simple ones to think about is salt, which is usually sodium chloride. And so there's some receptors that can just let the salt directly into the cell as a way to let that sodium in to start changing the ion content of the cell to change its ability to release neurotransmitter. Cranial nerves 7, 9, and 10 join together and form the main gustatory nerve, the solitary tract, it's called. The gustatory region is in the insula, right here, between the temporal lobe and the frontal lobe. Inside that cavity, there's an area called the insula on the inside wall here, which is dedicated to taste. And the gustatory pathways also direct information to the primary somatosensory region, helping us to identify the texture of our food and localizing taste and food on the tongue. There's also a branch of the nerve that goes to the orbital frontal cortex, where a mixture of our olfaction and our gustatory information occurs, which helps us identify the flavor of things, which needs both their smell and taste together. So next we'll talk about the neuroanatomy of motivated behavior more specifically, and focus on the hypothalamus and the pituitary gland, and how the hypothalamus can modify our body's functioning by changing activity in the pituitary gland. We'll also talk about the limbic system and the frontal lobes and how they can modify this hypothalamic control. We'll think about the idea of a homeostatic mechanism. Like our thermostats in our houses, these are processes that our body utilizes to maintain critical functions within narrow ranges, like our temperature or salt content or sugar content of our blood. The frontal lobe and limbic system aim to control our behavior through motivation based off our plans and our emotions, and they funnel this motivated behavior through the hypothalamus. The hypothalamus sends its axons into the brain stem, for example, to control circuits that can produce motivated behaviors in turn. And it also releases chemicals into our blood that can change our activation. Not all our behaviors are controlled via this hypothalamus. And so if I get you to just tap your finger on the table, that's kind of a non-hypothalamic task once it starts. Getting you to actually start doing it would need some sort of motivation that would involve the hypothalamus. It's primarily motivated behavior, therefore, that necessitates this involvement of the hypothalamic system. A regulatory behavior is a behavior that is motivated to meet the actual physical survival needs of an animal. So these are controlled by homeostatic mechanisms that include the hypothalamus as part of the circuit. Some of these are the body temperature, the eating and drinking, salt consumption, and waste elimination from the animal. Alternatively, non-regulatory behaviors are not necessary to basic survival of the animal itself. So they're not controlled by these same homeostatic mechanisms, but however, most involve the frontal lobes more than the hypothalamus. And they're strongly influenced by external stimuli. Examples of these non-regulatory behaviors that don't directly control our ability to be alive are sexual behavior, parenting behavior, aggression, food preference, our curiosity for certain things, and even reading is a good example. So here we can see this alternative types of behaviors in a chart. The hypothalamus aims to maintain homeostasis by modulating activity both of the endocrine system, which releases hormones into our blood, and the autonomic nervous system, which controls the functioning of our internal organs. It influences behaviors based off selections by the limbic system of what we should and shouldn't do, based off both past reward and based off emotion. The hypothalamic circuit controls many different motivated behaviors, ranging from how fast our heart is beating to our sexual activity. So the hypothalamic circuit controls hormone secretions, and it does this by controlling the pituitary gland, which is just below it in the brain and connected to the bloodstream. The pituitary gland is a gland attached to the bottom of the hypothalamus. It secretes chemicals into the blood that controls the activity of other glands throughout our body, like our lymph nodes. Sorry, like our thyroid glands here, not lymph nodes. And it's known to be associated with our biological rhythms as well. It can send and receive signals about our daily sleep and wake cycles. The hypothalamus can be thought of as having three regions, which we'll kind of see on the next image as well. The lateral regions towards the ears, the medial regions towards the center of the head, and the paraventricular region, even more central, around the outside of the ventricles. The paraventricular region, for instance, contains and releases oxytocin, which is a chemical that we know to be related to nurturing and pair bonding. So here's some images of the hypothalamus, both on a view from the inside of the brain, from the inner surface, showing this kind of grouping of different cells arranged this way. Here's the paraventricular nucleus. Here are the medial parts of the thalamus, the medial hypothalamus, sorry, and the posterior nucleus. We can see on a coronal section of the brain these same regions, the third ventricle, surrounded by the paraventricular regions of the hypothalamus. We can see here the medial hypothalamus right here. And then what's not shown on this image is the lateral hypothalamus here as well. And so different parts of the hypothalamus are involved in slightly different circuits. We'll see, for instance, that the lateral hypothalamus is involved in this something called the medial forebrain bundle. This group of axons that travels from the brainstem through the hypothalamus here onto the frontal lobe. So this lateral region contains both nuclei, but also axon nerve tracts that connect the brainstem to the forebrain, this area, the medial forebrain bundle. So it connects the brainstem with the limbic system. It forms the projections from the brainstem to the basal ganglia and the frontal cortex, which we've already learned about when we learned about the dopamine system and other neurotransmitter systems. For instance, the dopamine containing fibers here are involved in reward and contribute to our motivating behaviors. So here's that medial forebrain bundle traveling through the hypothalamus right here and connecting the brainstem to all these frontal regions. The hypothalamic hormone secretion pathway involves control of the pituitary gland. And the posterior and anterior parts of the pituitary gland are controlled separately by the hypothalamus in different ways. The posterior pituitary gland, closer to the back of my head, contains neural tissue. It's a continuation of the axons of the hypothalamus. And so neurochemicals get released directly into the pituitary gland there by the axons. The anterior pituitary gland is more like a endocrine, sorry, it's more like a gland. It's not part of the brain. It synthesizes various hormones there, and it receives signals from blood from the hypothalamus. The hypothalamus sends signals to the anterior pituitary through these releasing hormones. And so it releases hormones into the blood, and the anterior pituitary receives them and decides how much or how little of a hormone to release into the blood of the body. The hypothalamus can make peptides like oxytocin that are then transported down the axons to the terminals of the posterior pituitary first. And so this first technique is that the hypothalamus can make these chemicals and send them down the axons and release them directly into the posterior pituitary gland. And blood capillaries in the posterior pituitary can pick up these peptides and transport them into the bloodstream. And so, for instance, the oxytocin can be released based off the sound of a baby crying. And this system can then send oxytocin into the body that then can produce milk. And more generally in the brain can contribute to affiliative behavior and paired bonding. Here's some of that shown more clearly in a diagram. One here shows that oxytocin hormones being synthesized and sent down the axons to the posterior pituitary gland, where they're released by axon terminals and picked up by the capillaries and taken directly into the bloodstream. So blood comes in, it gets mixed with those chemicals and goes out, and the chemicals are released by these axons. Alternatively, the more posterior part of the hypothalamus releases these releasing hormones, and they're secreted into capillaries. And these capillaries connect into the anterior pituitary gland. These are not axonal connections, and this is not therefore part of the brain. And these capillaries release those releasing hormones, and these adjust the anterior pituitary cells' amount of hormone secretion into this blood. So blood's coming in here, it's getting mixed with these releasing hormones. The releasing hormones are deciding how much of the hormones to secrete out into the blood. And so by controlling these two types of circuits, the hypothalamus can control how much hormones are put into the blood at different times. So the hypothalamus also can, as we saw, release these releasing hormones, which can enter the blood and control the anterior pituitary. So the anterior pituitary produces a hormone called prolactin. And one example of this system is that its release is controlled by a prolactin releasing factor and a prolactin release inhibiting factor, which are both synthesized in the hypothalamus and released into the blood. And then prolactin can be released here depending on the ratio of those two. So these are some of the major hormones produced in the anterior pituitary as opposed to produced in the hypothalamus and transported to the posterior pituitary. Some of the ones that you may have heard about are growth hormone and adrenocorticotropic hormone, ACTH, which is important in our release of adrenaline. So here's another show of those two systems that can release hormones into the bloodstream. So what controls how much hormone the hypothalamus decides to release? Well, a number of things. There's feedback loops. So the chemical or the byproduct of the chemical can be detected by the body, and that signal can be sent back to the hypothalamus to clamp how much is being released once too much effect has occurred. Neural regulation can occur, so some brain regions can influence the hypothalamus to change the release of certain chemicals, like the sound of the baby crying, for example, we just covered, or the experiential response, like over time, these circuits can be modified by learning, and the amount of a chemical release can change given the same situation. So the hormones that are ultimately released by this system can go back and influence the hypothalamus to decrease or increase the secretions of those same controlling hormones, creating this feedback loop. And then, as we saw, these excitatory and inhibitory influences from other parts of the brain can also influence activity in these regions. So here's these two types of control. The target organs release hormones that can both act on the body, but also feed back to the hypothalamus, increasing or decreasing the release of those same hormones into the system. Or things like the sound of an infant or anxiety can modulate activity of the hypothalamus through connections, which can then change its release of something like oxytocin. And this is important for things like breastfeeding to understand. Just to be more specific, you can see how the ability of a mom to successfully produce milk can be related not just to the external stimuli like their baby being there and doing the right thing, but also to something like anxiety, which can decrease the release of oxytocin. If you electrically stimulate nuclei of the hypothalamus, you interestingly can produce goal-directed behaviors that are quite complex, like eating or drinking or digging, or fear responses like the cat arching its back, or reproductive behavior. Here you can see without stimulation, the animal just sits there, and once stimulation occurs in the hypothalamus, the animal vigorously digs. When it stops, it stops. Let's think next about the control of regulatory behavior, more specifically, not just the neuroanatomy. So regulatory behaviors maintain these vital body systems, like the amount of fluid in our body. And conversely, the non-regulatory behaviors that we aren't covering here aren't controlled by these same homeostatic mechanisms. So we'll talk about eating and thirst a little bit. Eating disorders can lead to being both overweight or underweight if you're eating too little or too many calories compared to how many you're expending. Obesity is defined here as an excessive accumulation of body fat due to overeating. And anorexia nervosa is a disorder in which there's an exaggerated concern about being overweight that can lead to inadequate food intake excessive exercising, and can lead to severe weight loss and starvation in extreme cases. Our digestive system is controlled by the enteric nervous system we covered, a set of neurons that live inside our stomach and intestines. There's detectors cells there that keep track of the levels of different nutrients in the blood. And the three main types of nutrients that are extracted by our stomach and intestines all have detectors in this system. And each represents a specialized form of energy that our body uses. We have lipids that we need, like fats. We need amino acids from things like beans and meat, which are the building blocks of proteins. And we need glucose for our brains, sugar. So when we eat, the food travels from our oral cavity through our esophagus into our stomach, where hydrochloric acid begins to break it into small particles. And the enzyme pepsin can break down the proteins, like things like meat and beans, break them up into the amino acids. The food then moves through the duodenum to the upper small intestine, where it's broken down further, allowing the bloodstream to absorb amino acids, fats, and sugars. Finally, the large intestine absorbs water and salt from the food, and the waste passes out. Our ability to eat and our motivation to eat seems to be controlled by our brain, and we can have two disorders on opposite ends of this called aphasia and hyperphagia. Aphasia is a failure to eat. It might be due to unwillingness or unmotivation, or to motor difficulties like swallowing problems. The unwillingness situation occurs following lesions to the lateral hypothalamus, and electrical stimulation can elicit eating. Hyperphagia is the opposite. It's a disorder where an animal keeps eating and doesn't stop. It's observed following lesions to the ventromedial hypothalamus, alternatively, where electrical stimulation alternatively can inhibit feeding. So increasing activity of the ventromedial hypothalamus makes you eat. Increasing activity of the lateral hypothalamus, increasing activity of the ventromedial hypothalamus stops you from eating. Increasing activity of the lateral hypothalamus makes you more likely to eat. When I say increasing activity, I'm talking about this electrical stimulation. Damage to these areas and to the paraventricular nucleus could have multiple effects. Can change your hormone levels like insulin. It can lead to changes in your reactivity to food and how good food looks. It can interrupt your ability to modulate your amount of glucose and lipids in the blood and your metabolic rate, leading to changes in those levels. These systems are controlled in part by systems in the brain like the amygdala and the orbital prefrontal cortex. The amygdala projects directly to the hypothalamus, and damage to the amygdala can alter food preferences and can also abolish taste aversion that's been learned. The orbital prefrontal cortex we saw receives input from both the olfactory and gustatory system and is related to our sense of taste. Damage can result in decreased eating because of the same food doesn't look and smell as good as it used to. Next, we'll talk about drinking. So there's actually two types of thirst. There's osmotic thirst. This results from an increased concentration of chemicals like salt in the fluid. And we need to drink more pure water to restore that concentration. So we have way too much, say you've eaten way too many chips. What you're thirsty for after eating way too many chips to dilute the salt content of your fluid in your body is pure water. Alternatively, when your body loses fluid, like you've run in a marathon or a bike race, this is a reduction in fluid volume overall. And if you were to just drink pure water, you would end up having way too low of chemicals in your blood because you would dilute them so much with that water because of how little fluid is left. And so in hypovolemic thirst, we're encouraged both by our brain, and I'm encouraging you to drink fluids other than water, like Gatorade, to restore those electrolytes as well. So osmotic thirst we saw is a deviation from the ideal solute concentration, the amount of salt and other chemicals concentrated in the water, and it can activate a system to reestablish that balance. There's receptors in the hypothalamus along the ventricle that detect the amount of solute in the body fluid, and they send a signal to the hypothalamus to stimulate us to drink. If we drink too much, our body tissue can swell with the excess fluid, and this can drown our cells in fresh water. This can create something called water intoxication. And this can occur, for example, if you have hypovolemic thirst and you just drink pure water. So if you run a marathon and then you drink too much water without any electrolytes, your body tissue will have too much water and not enough chemicals, and can lead to this intoxicated feeling. Hypovolemic thirst arises when the total volume of body fluid declines. We need to drink flavored beverages that contain salts and other nutrients when this happens, and we actually get an urge, as opposed to an urge to just drinking water, to drink more complicated drinks. The kidneys can detect these changes, and they send a hormone signal, angiotensin, that stimulates the midline hypothalamic neurons to motivate us to drink these tasty drinks. Next, we'll talk about control of sexual behavior and sex differences. First, we'll think about the major differences between the sexes in animals, and they're displayed in the textbook in this chart. Hormones cause sex differences by acting during development, but continue to act during adulthood. There's sex differences in behavior of animals. There's sex differences in physiology of animals. There's sex differences in the susceptibility to diseases. There's sex differences in neural and glial structures and connectivities, in neurochemistry. We'll see today how the androgens and estrogens play a role in sexual differentiation in the brain, and how the sex chromosomes that we have contribute to sexual differentiation a little bit. We'll see a little bit about this, and we'll see that the sex differences are context dependent, and that sexual differentiation occurs due to 4 key processes: neurogenesis, cell migration, cell death, and the differentiation of certain circuits. Won't necessarily cover all those topics. They're major kind of assumptions related to this study. So hormones during brain organization have 2 effects. During development, they organize the brain in certain ways. So our gonadal hormones like estrogen and testosterone organize the brain to be in a particular structure. And then in adulthood, once it's that way, they activate certain sex-specific behaviors, such as mating behaviors. So these chemicals have both organizing and activating effects. Many of our behaviors in animals show sex differences. And in humans, for instance, female children show verbal fluency advantages, and males and females show differences in their spatial behavior in the types of cues they rely on average to navigate. These differences aren't absolute. They're both on a continuum, but they're also can change, and they're likely related to sex differences in the brain due to the organization and activation. The differentiation is also related to the genes on our sex chromosomes. Those with Y chromosomes have different genes that can express in different ways, causing different differentiation. Sexual dimorphism refers to the difference in development between the two sexes. And as we've seen, these are influenced by the gonadal hormones testosterone and estradiol. Counterintuitively, it's estradiol that masculinizes the male brain. There's a chemical called aromatase, which converts testosterone into estradiol. And the estrogen is prevented from entering neurons itself. And so it's only this estradiol in the brain that can modify the brain circuits of the males. And this modification, one effect is that the preoptic area of the hypothalamus is 5 times as large on average in males than in females. So that's an organizing effect. An example of an activating effect of sex hormone is that the female sexual behavior and other behaviors can vary across the estrous cycle. So the estrous cycle is associated with changes in estrogen levels, and high estrogen levels are associated with sexual receptivity in females. So rats that are high in estrogen also have been shown to have more dendritic spines on their neurons. So their neurons actually cycle every 4 days of their estrogen cycle between having more or less spines. In adult males, for example, testosterone levels can influence motivation to seek sexual behavior and also more acutely affect the production of copulatory behavior. Here's an example showing those differences in the cycle of a rat in the number of spines on the dendrites between day 1 and day 2 or 3 of their estrogen cycle. Our sexual behavior is controlled by a number of these brain regions, and this has been understood in studies of animals, for example. The ventromedial hypothalamus can control a female's allows the ability of the female to control her mating posture, to be receptive to mating. The preoptic area of the medial hypothalamus, which we saw was 5 times bigger in males, controls copulatory behavior, but not sexual motivation. And the amygdala, however, seems to be controlling sexual motivation in males and possibly females when outside of the estrous cycle. So we saw that several of these nuclei are 2 or 3 times larger in males than females. And this differentiation of the brain generally results from the effect of testosterone, which is complete by birth. And some of these differences in the hypothalamus that are creating these large differences in brain structure seem to be related to methylation of genes. And so certain genes are turned on and off by testosterone in development that leads to different brain structure. And there's been shown to be differences in hypothalamic structure of heterosexual and homosexually inclined men as well, relating to this differentiation of testosterone. There's several biological factors that appear to influence transgender identity as well, like variations in chromosome. There's polymorphisms of genes for estrogen and androgen receptors that seem to be related to transgender identity, variations in gonadal hormone levels, and also immune system activity directed towards the Y chromosome seem to be factors related to someone's identity, someone's gender identity. So in addition to just these lower level circuits like the amygdala and the hypothalamus, the cortex must also have a role in controlling our sexual behavior, but it's poorly understood. But we know this because damage to the frontal lobes, for instance, can result in a loss of inhibition about sexual behavior on one case, or also a loss of libido, a loss of sexual interest as well. Next, we're going to talk about the neural control of emotion. And I want to also add that you can go on and take courses about these topics in our department and across campus. You can take courses like hormones and behavior. You can take courses about emotion and motivation as well. So our emotion is a difficult thing to explain and study, and some ways of trying to study it we'll talk about today are dividing it into different components. So we can think about the autonomic response to of an emotion, like our heart rate increasing, our palms getting sweaty, things that you could measure with equipment. And we think about the hypothalamus and structures as well as the enteric nervous system that might be associated with these autonomic responses. Like I have a bad feeling in my gut about this. We have subjective feelings associated with those autonomic responses that seem to be related to the amygdala and frontal lobes. And then we even have cognitions about those feelings. We have thoughts about the feelings, like, I've been sad all day. And these occur in our cerebral cortex and our frontal lobes. One idea called the James Lange theory of emotion, after the individuals who proposed it, is that things like trembling and rapid heartbeat that are associated with emotions come first, and our brain interprets these as an emotion given the situation that we're in. Our cortex then produces cognitive responses to this autonomic information. And so this way, our gut activity could be interpreted in some way by our brain. One evidence for this is that people with spinal cord injury, that leads them to have a decreased reception of sensation from their internal organs, right? Because the sensation from our internal organs is transferred into the sensory receptors in our spinal cord and up to our brain. The higher up your spinal cord injury means that you receive less and less internal organ information. And they showed that the more someone had a higher and higher their spinal cord injury, the more they had a decrease in emotionality following the injury. And so this was proposed to show that the less you have these bodily feelings, the less you experience emotion. A related idea called appraisal theory thinks about emotion more as a process that you're in rather than a state that you're in, a process that you're doing, sorry. And they think about emotion as activity of several systems or components together. There's an appraisal component in which you evaluate the context. You're like, oh, I'm in a classroom and everyone seems to be writing a test. There's a somatic component, the physiological effect, like the sweatiness and the heartbeat, a behavioral component, and then a feeling component, the subjective experience. Emotion seems to involve this limbic cortex, involving the cingulate and the hippocampus as well. These areas are called allocortex. They have, instead of 6 layers, they have 3 or 4 layers of cortex. And we'll talk about the hippocampus more in a couple of weeks when we talk about learning and memory, but it's in the medial temporal part of the temporal lobe here. It takes part in things like memory and spatial navigation, and we saw last week it's vulnerable to stress. And the parahippocampal cortex is area adjacent to that hippocampus. Broca, whose Broca's name is after, Broca's area is named after, originally proposed this idea of the limbic system, including the cingulate gyrus, the hippocampus, the amygdala, the olfactory system, as well as the hypothalamus and the thalamus. And you can see that's kind of a big grab bag of different areas, but we can still think of the hypothalamus as playing this important role in the output of behavior, as we've seen, involving this emotional system. There's a circuit here called the PES circuit, in which information from the frontal lobe and the somatosensory system all ends up funneling in to the hypothalamus before it gets output into our motor system, showing this idea of this funneling of various systems into the hypothalamus that produces our emotion and motivated behavior. A circuit diagram of A shown in B shows some of that idea here, showing the hypothalamus as the output to behavior, which receives input from the amygdala, the hippocampus, and the prefrontal cortex, as well as the mammillary nucleus adjacent to it, and then creates these circuits that can feed back to modulate its own behavior. And so the idea is that the emotionality of a stimulus, like your memory for how fearful that is, can influence your likelihood of behaving in a certain way. Most of these limbic structures, like the amygdala and hypothalamus, take part in emotional behaviors. And they also have important roles in our motivated behavior, such as especially species typical behavior like feeding and sexual activity. The amygdala is an almond-shaped collection of nuclei in the limbic system at the end of the hippocampus. It plays a role in emotional behaviors. As we've seen, it receives input from all the sensory systems, so it can find out what might be scaring us. Many of the neurons in the amygdala respond to more than one sensory modality. They might respond to the sight and sound of a cat, and they send projections to the hypothalamus and the brainstem on to control behavior, and they're intimately connected to the frontal lobes as well. And so the amygdala we can think of as involving conscious awareness. Oh, it involves the autonomic and hormonal responses by its connection to the hypothalamus. And it seems to influence our conscious awareness of the consequences of events via its connections to the prefrontal cortex. So we can see like, this is bad, and I know it's bad because of that thing I did. A syndrome called Kluver-Bucy syndrome, where the amygdala of a monkey was removed, led to an amazing set of symptoms like tameness and a loss of fear, indiscriminate eating, crazy sexual behavior, increased sexual behavior, not crazy, but kind of a dampening of inhibition of sexual behavior, tendency to react and attend to every visual stimulus, tendency to examine things by mouth, and an inability to recognize objects. So the amygdala therefore seems quite important for survival of an animal. The electrical stimulation can produce autonomic responses like changes in blood pressure and arousal, as well as feelings of fear, indicating its tight connections into the hypothalamus and our body. And our olfactory system, we saw, can connect directly to the amygdala, so it can directly activate our sense of fear in this way. It influences the hormonal responses and our conscious awareness, we saw. The prefrontal cortex, similarly, contributes to our emotion by specifying the goals of why we're moving. And it selects certain movements that are appropriate for a given time and context. And the context might be internal, like, oh, I gotta get out of bed. Or it might be external, like an alarm clock. Or it might be a distant memory or an emotion, or responds to some context. And so our frontal lobe has to decide what to do when. Which of your homework to study first, and has to base this information off things like emotion and memory. The orbital frontal area of the prefrontal cortex has direct connections to the amygdala and hypothalamus. And when you stimulate it, it can also produce autonomic responses via these connections. Damage to the orbital frontal cortex can produce personality changes like apathy, loss of initiative, and drive. It seems to be responsible for awareness of the emotional states that the rest of the limbic system produces. And so the system produces some state, and then the prefrontal cortex can kind of watch that happening and report it to us. The prefrontal lobes are divided up grossly here into the dorsolateral prefrontal cortex, premotor cortex, orbital frontal cortex, ventromedial prefrontal cortex, and the anterior cingulate cortex. We're focusing here on this orbital frontal cortex and the ventromedial prefrontal cortex. Generally, this big set of brain areas is very poorly understood. And in the future, in 10, 20 years, maybe there'll be whole chapters of this textbook about each of these regions, understanding their different roles. But right now, we're talking about them quite generally. The prefrontal cortex receives this very highly processed information from all our sensory areas. It's the top of the hierarchy. Your visual system, your auditory system, your somatosensory system sends all its information to the prefrontal cortex. The neurons there are multimodal generally. It receives dopaminergic input from the brain stem via that more medial forebrain bundle. And the axons that output from the prefrontal cortex can influence the autonomic and enteric nervous systems to control our eating and drink, blood pressure and respiration, for instance. So we have all this input from the thalamus, from sensory areas, from the dopamine areas, and from the amygdala. And then we have output to the basal ganglia, the premotor cortex, the hypothalamus, to produce behavior. Damage to the prefrontal cortex can have severe effects on social and emotional behavior, like inability to express your emotions or recognize other people's emotions, apathy and loss of drive, and especially damaging is inability to plan and organize, which leads to very poor decision making. For many years in the 50s and 60s, a procedure was popularized that was quite dangerous and ineffective in the end, called a frontal lobotomy, in which people's frontal lobe was lesioned through their eye cavity in this way, as a way to try to decrease their behavior that was unwanted. And this was originally thought to just make people more relaxed, but it was quickly understood that it led to these long-term planning and decision-making difficulties. Major depression is a disorder characterized by prolonged feelings of worthlessness and guilt, disruption of eating, disruption of sleep, slowing of behavior, and thoughts of suicide or death. It has a genetic component, and it's biological abnormality. So we can think about it as a problem with our physiology. So for instance, early life stress can produce epigenetic changes in the prefrontal cortex that can lead to these lasting abnormalities and circuits. Some other emotional disorders we can learn about, and you can learn about in other classes in the department, are generalized anxiety disorder, which is these persistent high levels of anxiety, which are often accompanied by maladaptive behaviors aimed to reduce those anxieties. Generalized anxiety disorder is thought to be caused by chronic stress. Phobias are dreaded objects or greatly feared situations that are clearly defined, like agoraphobia, fear of going out in public, or arachnophobia, fear of spiders. Panic disorders are recurrent attacks of intense terror that come without warning and without any relation to external events. For all three of these, the most common treatment is benzodiazepines, which can increase GABA's effect, just like having a drink of alcohol. And you can imagine they're similarly addictive. Finally, we'll talk about reward. Reward is one of the other motivated factors in our behavior. We're more likely to do things that led to rewards in the past, and we're less likely to do things that led to punishment. So our reward mechanisms likely evolved to increase our fitness as a species and as individual members of the species. We want to search out and take and be with more of things that lead to reward, and we want to avoid things that lead to punishment and make us less likely to succeed. Olds and Milner in the 1950s found that they could make a rat press a bar that electrically stimulates certain sites in its brain, called intracranial self-stimulation. And the lateral hypothalamus and the medial forebrain bundle, which are part of that dopamine system, are especially effective when you use them in this self-stimulation task. So a rat will press a button to send an electric shock into its medial forebrain bundle, and therefore we interpret that as being a pleasurable experience. So here's that dopamine system going through the medial forebrain bundle to the prefrontal cortex. So when an animal is engaged in this intracranial self-stimulation, dopamine is released. Drugs that enhance dopamine increase self-stimulation. Drugs that reduce dopamine reduce it. And therefore, we can interpret that the amount of dopamine release seems to be related to how rewarding an event is. When animals engage in a behavior like feeding or sex, dopamine release is increased all over these circuits. And as the animal learns to associate certain cues with upcoming food, dopamine becomes released by those cues, which we'll see in the chapter on learning. Highly addictive drugs like nicotine and cocaine increase dopamine levels in the nucleus accumbens, which is one of the reasons why they're thought to be so addictive. It's similar to this self-stimulation, but with the drug in the middle. We believe that reward can be better thought of as two independent processes, wanting and liking, which we talked about last week. Wanting is the incentive and liking is the evaluation of the pleasure. So they don't always occur together. And when they don't occur together, it's often bad. So when one wants more than when one likes. Wanting seems to involve this dopamine system. So changes in dopamine are associated with us wanting some things and not others, whereas liking seems to involve the opioid and GABAergic systems. A rat with electrode in the lateral hypothalamus will eat when the stimulation is on. And even though it's eating, when you look at its face, it looks like it's not enjoying the food. And so it has this aversive response to eating, even though it's eating and eating and eating. So it increases the wanting for the food, but not the actual liking experience of the food. You can also think about people outside in -20 in the snowstorm smoking a cigarette. Don't look like they're enjoying themselves very much. So a reward system processes wanting and liking independently of one another. And we can think about the dopaminergic system, wanting system activating, but also this liking system involving the brain stem and prefrontal cortex associated with opiates and the actual pleasurable experience. This leads us to propose that there could be this kind of pleasure electrodes. We could implant electrodes in those pleasure areas and experience that pleasurable experience of liking beyond just the wanting. We can think about pleasure as a kind of a gloss, which creates this pleasure effect. It's a gloss of objects or events, and it's generated in the activity of the brain by some balance of how much activity there is in these red regions on the map that I showed you on the previous slide. So in the past decade, some deep brain stimulation techniques have been developed, and it could be possible to try to stimulate with electrodes in these areas to try to increase people's happiness without modulating their dopaminergic system. So that brings us to the end of this lecture. Next week, we're going to talk about sleep